Have a wonderful day, everyone. I am the leader of the group 4. I am Rosemary Gituayan Fantes, and this is our experiment number 3, which is protein. But before that, let me allow to introduce my member first. So, yours truly, Fantes Rosemary, Kamsa Sarah Jane, Giali Armira, Bangkas Jimnaila. And also I conclude Shena Datukan. The introductions of the experiment 3, which is protein, are polypeptide structures consisting of one or more long chain of amino acid residues. They carry out a wide variety of organism functions, including the DNA replication, transporting molecules, catalyzing metabolic reaction, and providing structural support to cells. And now, I proceed to the objectives of the experiment. So first objective, which is observe and record the result on the data provided below. Number 2, determine the certain characteristic group detected using the different chemical. Number 3, at the end of the experiment, students must participate on what we given task. Another is safety data. First is substance name, which are igiok NH23 acetone. Formula RCH, open parenthesis, CH2, close parenthesis, COOH. Synonyms, polypeptide. GHS pictogram. Symptoms related to the physical, chemical, and symptoms related to the physical, chemical, and toxicological characteristics. For the signal word of the experiment, wait until the color can change. Hazard statement, do not try this at home. Chemical reaction can be very dangerous. Precautionary statement, add, mix, and heat the test tube. Disposal, always keep the chemical spill. Appearance, different type of chemical substance have identified in blue, white, and orange. Color, more on blue and white, also yellow. Odor, like odor of egg. pH, which is 4. Melting point, 3 to 5 minutes of heating. Boiling point, 3 to 5 minutes also. Flash point, 3 to 5 minutes. Flammability, hazardous chemical. And now, I proceed to the data and result of the experiment given. First data and result is coagulation of protein, which is they have test. See, they have heating H2O3 and ethanol. For observations of heating is generation of protein, have changes of color. Diluted for NH2O3, we have slightly no color changes, and for ethanol is generation of protein because they have changes of color happen. Letter B of data and result is precipitation by salt of heavy metals. So also we have test copper 2 sulfate. With observations is generation of protein also with color blue, and lead 2 acetate, which is generation of protein, which is whitish color. Color test for protein. We have test ninhydrin test. Observation, blue color, appearance of protein. Biorate test, blue color, appearance of protein. Xanthoprotic test is orange color, indicating of protein also. Millions test is phenol, tyrosine, and egg albumin have the appearance of protein, or they have changes of color into pink or red color, or what we call plus test. For Hopkins to call test is tryptophan. Sulfur test, we have changes of color, which is black color, the presence of sulfate. Question number 1. Draw a positive structure for the complex that gives the positive test result for the biorate test. So this is the structures of complex, which is they have positive test according to biorate test. So may peptide bond to biorate compound. So we can see they have the structures of biorate compound. For number 2 question, suggest a reason why the peptides do not give a positive test result with the biorate. So, hence look at your answer in number 1. So, for me, because polypeptide have a structure similar to biorate, They are able to complex with copper by the biuret reaction. Single amino acid and dipeptides do not give the biuret reaction, but tripeptides and larger polypeptides or protein will react to produce a light blue to violet that absorb light of 540 nm. Number 3 question. Suggest a reason why serine and threonine give positive test result with the biuret test. Look at your answer in number 1. So, I base another to the number 1 question or my answer in number 1 question. So, this is my answer. The reason is serine and threonine give positive result test with biuret test because histidine histidine is the only amino acid that give biuret test positive. Number 4 question. Egg white and milk are used as antidotes for heavy metal poisoning. Offer an explanation. So, for me, milk is not a remedy or antidote for poisons, nor does it protect the stomach from indigestive chemical or toxin. Other myths, including having a person eat burnt toast, raw egg, or mustards, none of these are remedy. Number 5 question. Both picric acid and tannic acid have been used in the treatment of burns. Offer an explanation. At the present time, the tannic acid treatment of burns is in more general use than any other. It has attained its great popularity through its success in extremely severe burns in which the life of the patient is at stake and would dressing form of an important part of the problem. Number 6. Why does nitric acid stain the skin a yellow color? Nitric acid is known to cause yellow stain when it comes in contact with human skin. This is mainly due to it reacting with a protein called keratin that is found in the human skin. This reaction is often termed as xanthoprotic reaction. Number 7. How does the xanthoprotic test differ from the Heller ring test? Xanthoprotic test is used to detect amino acids containing an aromatic nucleus or tyrosine, tryptophan, and phenylalanine in a protein solution which give yellow color. Nitro derivatives on heating with con NHO3, the aromatic benzene's ring undergoes nitration to give yellow color. 8 question. What is the only amino acid that contains a hydroxyphenyl group? Tyrosine is an alpha amino acid that is phenylalanine bearing a hydroxy substitute at position 4 on the phenyl ring. It has a role as a Dempnia magna metabolite rather. 9. What is the only amino acid that contains the indole group? Tryptophan is the only amino acid that contains the structure of an indole. Example, a bicyclic compound consisting of the 6-membrane benzene ring fused to a 5-member and containing pyrrole ring. For the last question, which is number 10, in the sulfur test, what is the formula of the colored precipitate? So, a positive test in the lead sulfide test is represented by the formation of black precipitate at the bottom of the test tube. This indicates the presence of cytine or cysteine in the solution. For methodology of the experiments, for our general methodology for the different experiments in protein is the secondary and tertiary structure of a protein can be disrupted by heating, by pH changes, by high concentration of substance such as urea, or by reducing agents that break the disulfide bonds. The process is called denaturation and it destroys the protein's biological activity. So these are general methodology about proteins, all of that experiments process. Lastly, discussion and conclusion. So for discussion and conclusion, When I also know that protein denaturation have the presence of protein, for example in heating process, H2O3, and also for acetone or ethanol, and what the precipitation of salt in heavy soil, they have changes of color in different color rather. Test of alkaloidal precipitate in heavy presence of protein in every source of chemical. That's all my observations and also conclusions in all the experiments made about the protein. And this is submitted to Macder Barbara Queen. And thank you for listening, ma'am. I'm so sorry for my voice because I'm not feeling well po for now, and no one can help me for the experiment na ginawa ko po. Thank you.